In this video, we're going to look at the most simple example of mutations and the potential consequences that these mutations can have on the formations of the proteins that they regulate. Now, point mutations are named this way because they are simply a mutation to a single nucleotide that usually results from some sort of error that takes place during DNA replication. For example, if we take a look at the native form of a DNA sequence, what we call the wild type form, we see that there is normally an A base on the top strand of DNA here, but as a result of a replication error, there is now a T base because of incorrect pairing by DNA polymerase 3 or DNA polymerase 1. However, we can see that this change is now permanent because T and A are supposed to pair together, but because of the error that occurred in DNA proofreading, it fixed the wrong side or the wrong strand of DNA, resulting in a permanent change, and this is an example of a point mutation. In other words, the A base has been substituted with a T base here, which is why we can also call point mutations substitution mutations. Now, depending on where this substitution mutation occurs, it can have a number of possible effects, ranging from no effect whatsoever to possibly catastrophically affecting the formation of a protein. So the first and probably most common example of point mutations are what we call silent mutations, the first of which we'll look at is here. So all of these point mutations here are all based on the original sequence, again, what we call the wild type sequence here of the DNA bases TTC, leading to a codon AAG, which in turn codes for the amino acid lysine, which is important to look at because it has a fairly distinctive positive charge on its R group. Now, in this case, the point mutation that we're going to make is substituting this C for T base here, resulting in a change to the codon from AAG to AAA. However, what we can see is that these two codons are what we call synonymous codons, meaning that even though they are spelled differently, the meaning of the codon, the amino acid that they code for, is exactly the same, which means that a silent mutation actually has no change whatsoever to the amino acid sequence because the codons being synonymous result in the same amino acid being laid down during translation, and therefore there is also no change that occurs to protein folding whatsoever. This is why geneticists believe that silent mutations are actually the most common type of mutation, because if they occur in an actual protein coding gene, you can theoretically have an unlimited number of silent mutations, and there would be zero effect on a protein, for better or for worse. However, in the two examples here, we see that there are actually changes that occur to the amino acid being expressed, and these are what we call missense mutations, although the potential effect of a missense mutation depends on what the substitution is. For example, if we substitute the middle T base for our DNA triplet, it results in a different codon being created, AGG, rather than AAG, which results in the amino acid arginine instead of lysine. However, like lysine, arginine is a basic R group, which means that it will have a positive charge, and because it is relatively the same size as lysine, arginine has very similar, if not the same, physical and chemical properties to the original amino acid that it is substituting, and therefore the change to the protein folding is likely going to be relatively insignificant compared to what the alternative is. Now, in the alternative here, we can see that now it's the middle base, the middle T here, that we are substituting for a G, resulting in the codon ACG being produced, which codes for the amino acid threonine. Now, threonine is a polar amino acid, but doesn't have an amine or an acid functional group, giving it no charge. Now, because of this, we can say that this amino acid has definite different properties compared to the original amino acid lysine, and as a consequence, the change in protein folding is going to be much more significant than a change to an amino acid with the same chemical and physical properties. To take a look at what the potential consequence of this here, this particular substitution was not random. It actually refers to a real documented mutation here, and we can illustrate how important the location of the mutation is because of how it connects to protein folding. So here we can see that a positively charged basic R group is forming an ionic bond with a negatively charged acidic R group, which causes this particular protein folding in the tertiary structure to happen here. However, if we take the basic R group that originally existed here and substitute it with an uncharged R group here, now the negative charge on the acidic amino acid here has nothing to attract to because there is no longer a positive charge here, and now the protein folding has changed relatively significantly. And for an even more extreme example, we can look at the original mutation that we have here. So the original amino acid is glutamic acid, which happens to be negatively charged, but as a consequence of this mutation changing what the codon is, this results in valine being put into the protein here, which not only isn't charged, it's nonpolar. And as a consequence of this single mutation, it can turn a red blood cell, which is normally a biconcave disc shape, into something that looks like this. And this is an actual medical condition called sickle cell anemia, where your red blood cells effectively develop curves that look like sickles, hence the name, on the side like this, which can have catastrophic consequences as far as blood flow goes, because the tendency of these red blood cells is to hook and stick to each other like this. So as the result of a single mutation, it can have catastrophic consequences depending on the nature of the change to the amino acid and where that change occurs. But the most extreme example we save till last, and these are what we call the nonsense mutations. And if we take a look at the example here, if we substitute the first T base for an A base here, this results in the creation of the codon UAG, which you should immediately recognize as one of the three possible stop codons. Now, this stop codon, on its own, a stop codon is not bad. It's necessary to terminate protein formation. However, this stop codon is supposed to be a lysine. In other words, we have created a premature stop codon that occurs before it is supposed to occur, and therefore causes the protein to terminate before it should. This means that all of the other amino acids that occur after this premature stop codon never make it into the protein at all, and the protein will probably not fold and therefore probably not function properly, which is why nonsense mutations very often result in catastrophic failures as far as the folding of proteins go. However, once again, the key is the location and severity of the mutation together determine how big the consequences are of the mutation's effect on the protein. In the next video, we're going to look at the more complicated form of mutations called frameshift mutations and why these particular mutations can have even more significant effects than any of our point mutations do.